In this topic, we're going to discuss aerobic respiration and the experiments involved in aerobic respiration. So we're going to look at what is aerobic respiration, the word equation, and if you're doing extended, you need to know the equation in symbols, how energy from respiration and energy from combustion are linked, a calorimeter, the equation to calculate the energy content of food, how to test for respiration, the experiments to test for carbon dioxide and heat released during respiration. So what is aerobic respiration? Well, this is the release of energy from the breakdown of food substances in the presence of oxygen. And this is called aerobic respiration because oxygen is used. And remember that it takes place in little organelles inside the cell called mitochondria. So single is mitochondrion. And these produce a large amount of energy. So respiration produces a large amount of energy if oxygen is present. So what is the word equation for aerobic respiration? If you asked to write down the word equation, think about what is being broken down and what is needed. So think about you need to eat something. So if you eat glucose, you need to breathe in oxygen. Together, these react inside your cells to produce carbon dioxide plus water plus energy. And notice how glucose is completely broken down to carbon dioxide and water in the presence of oxygen. Now, if you're doing extended, you need to know the equation in symbols. So it's a good idea to write down the words first and then add the symbols later. So glucose is C6H12O6 plus 6 oxygen forms 6 carbon dioxide plus 6 water plus energy. So again, notice how glucose is completely converted into carbon dioxide and water if oxygen is present. So how is energy from respiration and energy from combustion linked? Well, the release of energy in respiration is the same as that released during combustion in the presence of oxygen. So combustion is burning. So if food is burnt, it is oxidized to carbon dioxide and water, and energy is released as heat. So the heat given off when food is completely burnt will show how much energy was stored in the food. And if we use this energy to heat water, we can find out how much energy was stored in the food. So let's have a look at how this relates to the lab. Now, you might be asked as a scientist to investigate the energy content of a peanut. How would you go about this? Well, you know that when something burns, it releases energy that can be used to heat water. So that's what you're going to do. You're going to burn the peanut and see how it raises the temperature of water. So you use something called a calorimeter. So this simple apparatus here is a calorimeter, and it's used to find the change in temperature of the water when a peanut is completely burnt. Now, before you start the experiment, it's important to know the mass of the seed or the peanut in grams, the volume of water in cubic centimeters, and the start and end temperatures of the water in degrees Celsius. So the procedure is as follows. You weigh the seed or the peanut, then you attach it to the end of a mounted needle, and you add a certain volume, for example, 20 cubic centimeters of water to the boiling tube with a stirrer and thermometer. You record the temperature of the water, then you set the peanut or the seed alight by placing it in a flame for a few seconds. And you hold the burning seed or peanut under the boiling tube until it is completely burnt. And you've got to stir the water and record the temperature. So you're looking at the change in temperature from the start of the experiment to the end of the experiment. So here's a picture of a calorimeter that would be used in industry. Remember that calorimeters measure energy. So calories are the old units for energy. A proper calorimeter is insulated so no heat escapes to the environment. It's also got a stirrer to distribute the heat throughout the water. Now that you've found the rise in temperature of the water, how can you find the energy content of the peanut? Well, you would use this formula. Now, you don't need to memorize this formula. You just need to be able to understand how to use it. So it's volume of water times rise in temperature times 4.2 divided by mass of food times 1,000. And the unit of energy is usually joule, but in class we will use kilojoule. So how can you test for respiration? So if you remember the equation for respiration, you'll notice that the products are carbon dioxide plus water plus energy. So you can test to see if carbon dioxide is produced by either using lime water or hydrogen carbonate indicator. So lime water goes from clear to milky white if carbon dioxide is present. So you can see 3 test tubes here. On the left, there's no lime water, it's just water. In the middle, you've got lime water, and on the right, you've got lime water. Notice how on the right, more carbon dioxide is being bubbled through the lime water. So the color, it's a darker milky white compared to the middle. Now, it's a good idea to hold up these test tubes against a dark background so that you can see the color change. So the theory behind this is that lime water is actually calcium hydroxide, and it reacts with carbon dioxide to form calcium carbonate, which is basically chalk, you know, when you write on a board. And this calcium carbonate is insoluble in water, so it forms a milky white precipitate. You can also test for carbon dioxide using hydrogen carbonate indicator. This changes color in the presence of carbon dioxide. So when it's neutral, it's red. And as carbon dioxide is added, it dissolves in the indicator and forms a weak acid that changes the indicator to yellow. So if you look here, you can see that when carbon dioxide is present, it's yellow on the left. And in class, I held up the hydrogen carbonate indicator, and it was a pinky color, so reddy pinky color. So there's a few experiments that you need to know, and we're going to look at these in class, so make sure you understand the theory behind each one. The first is to show that carbon dioxide is released during respiration, and the second is to show that heat is released during respiration. So in the first experiment, you would set up 4 flasks like this. So any active organism will respire to make energy, and it also means that it's going to be making carbon dioxide. So we need to provide the organism with air that has no carbon dioxide in it. And then we need to test the air given off by the organism to see if carbon dioxide is produced. So to do this, you set up these 4 flasks as shown in the diagram. Can you think of a reason for each flask, or the substance or liquid in each flask? Well, if you look at the direction of the arrows, that's the direction of the airflow through the apparatus. So you can add a suction pump to draw the air in the right direction. So if you look at flask A, this has got potassium hydroxide. So this removes the carbon dioxide from the air. Flask B has got lime water, and this is to test the air for any remaining carbon dioxide. Then flask C has got lime water to test for carbon dioxide released during respiration. So what do you think will happen at the end of this experiment? Well, at the end of the experiment, you should see that the lime water in flask C turned cloudy white. This is to show that carbon dioxide was released during respiration. So the lime water in flask B should remain clear because no carbon dioxide is coming into it. The carbon dioxide has been removed by flask A. Instead of lime water in flask B and C, you could put hydrogen carbonate indicator. So why are they different colors? Well, in flask C, carbon dioxide is present, so the indicator turns yellow. In flask B, the hydrogen carbonate indicator remains a pink color because no carbon dioxide is present. It's been removed by the potassium hydroxide in flask A. Now you could also use lime water in flask A to remove the carbon dioxide. So then think about what changes will occur in each flask. Well, the lime water in flask A will turn cloudy white, whilst the lime water in flask B will remain clear, and in flask C it will turn cloudy white. This is because flask A has removed the carbon dioxide, so it's reacted there. Flask B, there should be no carbon dioxide, so it should remain clear. And then flask C has got carbon dioxide produced from respiration. And the last experiment is to test to see if heat is released during respiration. So for this experiment, we use germinating seeds that are placed in an insulated flask. So it's important to have insulation because this will stop heat escaping. So what you do is you take a large number of seeds and you germinate them by soaking them in water. So lots of seeds will make our experiment more reliable. Then you take half the seeds and you kill them by boiling them. So this will be your control. Now, to ensure that nothing respires, so you don't have any bacteria or fungi respiring on these seeds, you need to wash them in something called hypochlorite. So this kills any bacteria or fungi that may feed on the seeds and respire. So this makes our results more accurate. So having a large number and repeating the experiment makes our results more reliable. So this is how to set the apparatus up. Now you can see that you've got boiled seeds. These are your control. You need to ensure that you've got an equal number of seeds in the control as well as the test experiment, or the same mass in both flasks. So remember, when you're describing an experiment, always have the same conditions, but you don't just write the same conditions. You need to say the same mass of seeds, the same initial temperature, the same volume of flask. So you've got cotton wool at the top there. The cotton wool allows for gaseous exchange without any heat escaping. So you leave these seeds for about 3 days. Now this experiment would be more accurate if the flasks were placed upside down. This is because hot air rises, so it will be trapped at the top of the flask if it was upside down. And carbon dioxide sinks, so it'll be able to diffuse out of the cotton wool. So what results would you expect? Well, you'd expect to see a rise in temperature in the flask of germinating seeds, showing that heat is released during respiration. Okay, in summary, we looked at aerobic respiration, the word equation, and the equation in symbols. We looked at how energy from respiration and energy from combustion are linked, something called a calorimeter, and then using the information from the calorimeter, so the rise in temperature, to calculate the energy content of food. How to test for respiration, so looking at how to test for carbon dioxide. Remember that you use lime water and bicarbonate indicator. And then the test to see if heat is released during respiration. So it's using those germinating seeds in an insulated flask. And that concludes our lesson. The end.